Hello, and welcome to a revolutionary approach to UV-Vis measurements, SoloVPE Slope Spectroscopy, presented by American Pharmaceutical Review and sponsored by CTech, a Repligen company. My name is Mike Auerbach. I'm the Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator during today's event. This webinar will demonstrate the science of slope spectroscopy and why variable path length is better than fixed path length, an overview of the SoloVPE system and its benefits, and a review of SoloVPE implementation in multiple applications. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speakers. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Joe Ferraiolo is the Associate Director Bioanalytics for CTech, a Repligen company, in charge of the SoloVPE variable path length UV solution. He has been with the company for more than 20 years, with over 10 years of development and validation experience in analytical applications. He specializes in UV analysis and leads the development and commercialization of high-value products and flexible solutions that address critical steps in the production of biologics. Joe, welcome to the webinar.
Thanks, Mike, for that introduction. Today we're going to talk about the SoloVPE system, the biopharmaceutical gold standard for UV-Vis spectroscopy featuring slope spectroscopy. What we're going to show you has been a revolutionary approach to UV-Vis. A couple of the topics we're going to discuss today is how the variable path length extension called the VPE works, challenges with current UV spectroscopy primarily related to traditional UV, some applications and publications backing the technology, and then ending with some of our customer validation data for how they've used our technology to replace their current platform. The CTech variable path length extension is the analytics arm of Repligen. We will be looking to implement our technologies in current products over the coming years, but this primary presentation is going to focus on how the VPE technology works, primarily related to the biologics line. With using traditional UV spectroscopy, we have an issue with single fixed path length cells being used for the analysis. As you can see in the equation above, the concentration is the variable here, while the path length remains fixed. This brings a host of different issues related to the current technologies, primarily related to dilution error. And if the sample does not fit within the linear range of the spectrophotometer and path length being used, a dilution step must be applied to fit that sample within the range of what the spectrophotometer is able to measure. Further downstream applications increase in time being associated with that method, having to send samples out externally for analysis, and then having the manufacturing floor wait on those times to be reported back in order to know whether or not they can continue their process. As you can see, the current product workflow has a series of 7 steps that must be completed in order to gain the results achieved using the current process. The main issue with current traditional UV is dilution. Whether it's gravimetric dilutions or volumetric, each come with their own associated errors. So it's been our goal not to just introduce another technique similar to what UV has done, but to revolutionize how UV is able to be performed within the lab space. So it's been our mission to essentially take the former method using traditional UV and turn it into something very simplified for the user in a measure report format. So we're either removing the error-prone steps associated with traditional UV and then automating the majority of the rest of the method. This gains time to results, robustness, simplicity, where a trained scientist doesn't only have to use the system, but improves workflow and validation throughout the entire process. Our approach is this. we are allowing the path length to be the variable here. So concentrations is what's locked. It's what's not changing. And since the concentration doesn't need adjustment, we are allowing the VPE technology to adjust the path length and find the most linear regression within Beer-Lambert's law to calculate your OD reading and therefore concentration. So we're doing two things differently in our UV technique. The first is the adjustment of path length. You can see where the light emits from the bottom of the fiber optic component called the fibrette. The light path is determined by where the light emits to the bottom of the sample vessel. The second thing we are doing is we are changing the path length and generating multiple absorbance values to calculate a slope regression. So slope spectroscopy is no longer concerned with absolute absorbance. We are concerned about the change of absorbance over path length, and this allows us to plot multiple values. By doing this, we can assign an R-squared value to each linear slope regression that's generated by the software. So what we've now done is taken UV and turned it into a pass-fail measurement due to the validity of the R-squared value having 3 nines or higher. So the spectra that you see in the bottom graph actually represent another path length scan of data. And the top graph would be the evidence of that, where we section the wavelength of interest for the calculation. How the system works is by using an Agilent Carry 60 as our light source. So again, a UV technology. However, we're just using the carry to bring the light from the light source into the SoloVPE. So all the measurements will be made directly in the SoloVPE itself. Once start collect is pressed, that piece of fiber, the fiber optic component called the fibrette, will lower itself into sample and set 0 path length. We equate this to tearing a scale. That way, we know that the measurements will take place starting at the bottom of the vessel. The software will then change path lengths while scanning the wavelength of interest. And what the software is looking for is one absorbance, or as closest to one absorbance as it can find. Once it finds that one absorbance path length, it will then generate 10 data points worth of regression data to create the slope regression. The most intuitive feature about the software is the user does not have to preload path length steps or path length ranges within the software. The software is intuitive to know that if it detects a very high absorbance change from path length to path length, it's dealing with a very concentrated sample. Therefore, it will use very small path length steps. Other way around, if it detects very little absorbance change from path length to path length, it knows it has something very dilute, and therefore will auto-adjust to a very long path length step. Completely automated and requires no manual sample intervention from the user operating the system. Data acquisition time is less than a minute. And the evidence of the data now is a slope-generated report. So the equation that we use to calculate concentration now is the slope, which would be m, divided by the extinction coefficient, which is E, equals the concentration of the sample. So we've now introduced a way to guarantee linear data without dilution error in order to make the measurement. The reason for this is the SoloVPE has a path length range from 5 microns to 15 millimeters. And we could take path length steps in as small as a 5 micron step, which results in over 3,000 choices of path length to find linear Beer's law. I'd like to show you some evidence provided by our customer base in their success of using SoloVPE within their process. This paper was published by Bioprocess International by Bristol Myers here in New Jersey. Not only have they implemented the Solo for the manufacturing floor, but this publication lists all of their validation data and has become a template within the industry on how to validate slope-based methods for biologics. Obviously, they had comparable data. However, compared to their standard UV-Vis, the percent difference is substantially higher than what we claim for the Solo. Most importantly, they were able to take a process that was over 3 hours down to 30 minutes by using SoloVPE. The reason for this is they're no longer sending out samples for analysis, having those scientists perform multiple dilution steps, analyze the sample, and then report back the data. You can make real-time measurements and decisions right on the floor. Additionally, they've also calculated that annually they save 33 days per system compared to using traditional UV. This next publication was generated by Biologics in Raleigh, North Carolina. They were the first company to actually publish their financial savings and a formal risk assessment in the same publication, which equated to over 50% overall cost savings by using Solo compared to traditional UV. One of the reasons for this, obviously, is no more calibration of scales and the equipment, dilution time by the analyst. So return on investment is substantially quicker on implementing our technology over any other UV technology, and they save close to 2 months of time by implementing VPE technology over traditional UV. Last but not least, last year, the variable path length technology was written into the ASTM standard for general practices of techniques of UV-Vis quantitative analysis. This standard is only revised every 5 years, and we approach them with data and publications to ask if we could be introduced into the standard for the variable path length portion, having enough equivalency data over the last several years to prove that we are a comparable UV technology. And that's what they've been able to do. So now in this standard, there is a notation that says to avoid the dilution step, the instrument may contain an automatic system which will allow the adjustment of path length of the measured cell to optimize the measured absorbance. This is variable path length slope spectroscopy. So as you can see, from regulatory to our customer testimonials, we have a nice bookend to provide the industry with a very high level of confidence that not only will we be comparable to any current UV method that's being run, but you will also get the time and savings by using our technology by not having to introduce multiple dilution steps within the process. This data was given to us by our customer base that we've been working with during validation of slope-based methods within their companies. The purpose of this slide is to show you that regardless of company, regardless of concentration range, that the repeatability on this particular method is well under the plus or minus 2% that we specify. But more importantly, regardless of group, whether it's formulations or QC, we've never had a company not save at least 1 month of time implementing the SoloVPE into their process. Benefits: no more estimating and dilutions or manual calculations. Everything is automated. Methods can be locked down for GMP compliance, so there's no risk or error associated with the reporting the correct information. A very wide dynamic range. Again, having the path length range from 5 microns to 15 millimeters expands the dynamic range of the instrument and what can be accomplished per method and per application. We're going to save time. We're going to reduce the production hold, and we're going to improve reproducibility and increase sensitivity throughout the process related to slope values accompanied by a passing R-squared value of 3 nines or higher. Protein concentration is certainly the strongest area of the Solo, anywhere from 0.01 mg per mil up to 300 mg. Other areas that we're going to touch on in the coming slides will be polysorbate analysis, Planova integrity testing, known as the gold particle test, gene therapy applications such as DNA and RNA, and in the future, getting into antibody drug conjugates, ADC testing, and multi-component analysis. So The purpose of this is to show you that while the majority of our customers have been using this for protein concentrations over the years, that if there is a current UV application you are using, you can certainly implement slope spectroscopy to replace that current method and gain all of the time and benefits associated with the device. So when we take a step back and look at how this can be implemented throughout the commercial workflow in biologics, it's important to know which tool to use for the appropriate application. So we'll be discussing a couple of these applications and then ending with what's next for the SoloVPE and where else we can implement a variable path length technique within the biopharma process, which will ultimately lead us to the FlowVPE. The first application we're going to look at is polysorbate analysis. The current UV method utilizes 600 nanometers of wavelength, where there's no active chromophore. We determined that there's actually an active chromophore at 235 nanometers. The reason why the industry has never seen this before is because the pathlengths used are typically in microns, not millimeters or centimeters compared to traditional UV. So what we're able to do now is actually quantify a working standard of polysorbate material and use that OD reading to know exactly how much PS80 or 20 will be used for the formulation of the product. Additionally, once the protein is introduced to the sample, using slope ratios, we can now determine how much protein and how much polysorbate within the same sample. There are multiple publications established for this technique, and this happens just to be one of them that can be downloaded from our website. A little bit about nucleic acids. Again, another multi-wavelength method here. We have proteins, we have DNA, RNA, and we focus on both of those areas with our method development. However, when they are together, it's more of a blended curve, and how do we actually account for that? So the first application actually came from Amgen when they were replacing their nanodrop with the SoloVPE. The nanodrop was only capable of measuring up to 3 mg per mil, and the reason for that is it's still a fixed path length technology. So while the path length is smaller, you still have the same issues with most traditional fixed path length measurements, where at certain concentrations, there's no way to guarantee or provide linear data. The second was plasmid purity done with Pfizer, looking to replace their traditional UV method doing the DNA RNA measurement with the protein. So we're looking at an A260, A280 method, both implemented in their environments, have replaced both of the former technologies, and have proven substantially that it's a cleaner and more accurate way to make this measurement for this particular application. I would advise you to look into these publications that go into more detail about exactly how they developed the method and the evidence based on what they found during the sample measurements. So an example of that would be in the purity of nucleic acids, taking 25 different mixes of plasmid and insulin and then comparing them to the expected value. Once we had comparable results, we then ordered third-party material and had both sites, including CTech in Bridgewater and Pfizer, measure both series of samples to prove that even though site-to-site, SoloVPE to SoloVPE and analyst to analyst, we were still getting the same comparable results of less than plus or minus 2%. Next, we're going to talk about Planova integrity testing, or formerly known as the gold particle test. This publication was put together with Bristol Myers in Massachusetts, noting their studies and their project to move away from traditional UV over to SoloVPE. The issues with this particular method are twofold. One, the standard that's provided by the vendor requires dilution. It's far too concentrated to be measured neat in a 1 centimeter cuvette. So dilution has to occur in order to start the process. However, the tolerance range is plus or minus 15%. So before we've even started the measurement, we're already introducing error. We obviously don't have that issue with the SoloVPE because we're able to measure it in neat form. The second part is post-filter analysis, where you are looking at a very dilute sample. And the issues that traditional UV have is, since you're only being reported a single absorbance reading, the analyst has no way to determine if it's related to sample contribution or instrument noise. Because the SoloVPE is providing you an R-squared value, we can guarantee when the process is clean due to no presence of active chromophore. So this is a refinement of a method to streamline and take the guesswork out of any user intervention with relation to analyzing the data. Next, E. coli growth, the OD600 test. The challenges of this particular test are twofold. One, the test has to be performed in live time. Yes, typically some samples are frozen during this process. However, you're trying to chase a moving target by measuring at time points when you still have continued cell growth within the process. Our approach was to work with an organization, in this case, the BCC in Switzerland, who did comparison testing between a standard spectrophotometer and the SoloVPE. The standard spectrophotometer data represented in the top right graph shows that after the 2 hours of the process, it requires dilution. So the samples are far too concentrated and must be diluted back down into the range of the spectrophotometer and the 1 centimeter cell. The issues with this are obviously dilution error, but the continued cell growth within the process. Whereas the SoloVPE data on the bottom left, no dilution is necessary, and since all the measurements are made in live time, there's no skew to the results based on additional cell growth. The project goes into detail about how the method was set up, and furthermore, with the introduction of the FlowVPE technology, using kinetic software, this entire process can be automated, where the user would just enter the time points for which they would like to collect the data, press start on the system, and then come back at the end of the process to the completed run. Lastly, virus titer. So we're talking about the empty-full ratio between the 280 and 260 wavelengths. This has multiple implications on how this was formerly done within the industry, whether the analyst is using qPCR, ddPCR, TEM, AUC. We at CTech wanted to understand, can we actually see a difference of slope, meaning a change of absorbance at both wavelengths of interest when measuring a full sample compared to an empty sample? And what we found was universally we were able to do that. So our approach was, if we can understand the change of absorbance by using the molecular weights of the protein and the DNA and known extinction coefficients, it's possible to revise this into a truly UV method and report in vector genome over mil criteria. And what we were able to show with one of our clients is that compared to their qPCR and ELISA data, we were within 9% of their expected result. Now, this is quite a departure for us since we're used to very tight tolerances, and the empty full method has quite large tolerances, as you can see by the plus or minus 20 to 50%. However, what is undeniable is that we were able to take a nearly week-long process and test on the VPE technology and get results within 5 minutes. This is an area still being explored by our company. It's one of our top priorities. So I would think in 2021, there'll be additional publications available and evidence of this application. Our approach to implementing SoloVPE, and more importantly, slope spectroscopy into the industry, was to make it the platform method within pharma. Concentration determination is by far and arguably one of the most critical steps within the process. Here it is an example of some of our success stories where the largest companies that have been using SoloVPE for the better part of the last decade have validated multiple drug substance and products using our technology to replace traditional UV. Additionally, these methods have been transferred to contract research and contract manufacturers. We are in 90% of the top 50 pharma companies that have implemented SoloVPE. When I say implemented, I don't mean they own a system that happens to be on the bench. I'm referring to validated methods for which the evidence of that data is being reported to regulatory agencies. We had a choice to make once the SoloVPE gained its wide acceptance into the market, whether to go more towards the research end of the process or go more downstream, and we've chosen to go downstream. The FlowVPE technology is our inline solution. It operates exactly the same as the SoloVPE. However, this is meant to monitor processes, chromatography, UFDF, fill finish, mixing. It's an entirely stainless device with a flow cell. The idea with the FlowVPE is to be mounted directly onto the skid or put on a mobile cart that can be wheeled next to any process, and you plug in your lines connected to potentially, say, an ATCA, and then monitor in real time. We have now taken this technology and put it into a process where all the analyst has to do is monitor what is happening in real time during a process. There's no more starting and stopping processes, pulling samples for dilutions, waiting on those results to know whether to continue or not. You are looking at a screen and making real-time decisions on when to stop and when to start. So here's where both technologies fit in the biologics commercial manufacturing pipeline. Okay, and as you can see, it starts and finishes with our technology being used in multiple areas. This is to give you an understanding of where the majority of our customers are using the VPE technology, and knowing that obviously in this stage, we are talking about GMP environments, which is something that we have a lot of experience in due to our leverage in utilizing our own software package to achieve 21 Part 11 compliance. Another example of how the FlowVPE can be used, especially within the gene therapy area, is looking at titer determination over time. So again, you can see monitoring the entire process allows for concentration determination and when the diafiltration starts, no sample preparation, no user intervention, and you're making decisions in real time. Our goal from the beginning was to revolutionize how UV is currently done within the industry. In our opinion, there's been no significant changes to the technique in over 30 years. Companies have taken spectrophotometers, made them smaller. They have taken the pathlength cells and made them smaller. But all of them rely on fixed pathlength absorbance values as their OD to calculate anything. The slope spectroscopy method is the only method that exists for UV that guarantees linear data every time the user presses the Start Collect button. So we believe that we have achieved our goal in a measure report method that can be used throughout any UV application throughout the industry. I'd like to thank you all for attending, and we will open it up for questioning now.
Thanks, Joe, for a very informative presentation. I'd like to remind everyone that you can submit any questions you have for Joe in the Q&A box on your screen, and we'll get to as many as we can. Also, there are additional resources in the upper right corner of your screen that are available for download. So let's now move into our Q&A session. Take a look at our first question. So Joe, our first question is, can you please talk about the validation process and validation support that C Technology provides?
Sure. So we understand that even though we believe in our technology is a superior way to do any UV measurement, we're completely aware of how much time is required within organizations with current project demands and schedules to be met, that we're going to provide a lot of assistance in helping with the deployment of the technology. So I'm not just talking about the standard IQ/OQ and preventative maintenance services. I'm talking about working directly with our applications group to help define the method for which the device will be used, and also the subsequent documentation associated with it, such as standard operating procedures, validation protocols, and then finally the assistance with either working with each company's IT group to help validate the software for its own internal use, or providing a service which allows us to come in and validate the platform for the user itself.
Excellent. Thanks, Joe. Our next question is, can you please explain how you get from slope to percentage full for gene therapy products?
Sure. Over the coming months, I would say stay tuned to our website for additional announcements. We are in the process of patent pending information related to this technique. We will be making it publicly available, but I will tell you that turning this entire method over into a vector genome over mil calculation involves the following. Essentially, we need to have 6 pieces of information. So we're talking about 2 slope regressions, as you've seen on the presentation, for both wavelengths of interest. In this case, it would be 280 and 260. We would need the molecular weights of each material. And lastly, we will need the extinction coefficients for each wavelength. The challenges associated with this particular method relate primarily around the extinction coefficients. There's not a lot of information to pull from. The most popular source material has been the Somers paper, which, again, they are assumptions made, so it's a good starting point. But in our experience, the users that do not have extinction coefficients, we are able, based on the information that we collect, are able to essentially back calculate the extinction coefficients for both wavelengths using slope. How the calculation is done, again, stay tuned, but there's a little bit of insight of what's required in order to get the calculation.
Okay, terrific. Thanks, Joe. Our next question is, are you seeing any difference in variability by protein concentration, noting the 0.1 to 300 milligram per milliliter range?
Sure. For repeatability, the answer is no. We maintain our plus or minus 2% across the entire range for repeatability. Related to accuracy, the only way that you can guarantee accuracy related to a measurement is when you are testing in this traceable standard. So that is the only comfortable way that we feel is responsible to leverage accuracy claims versus any other technology, that they should be using a NIST traceable standard. And when that is done, we've managed to stay within the same tolerance range.
Excellent. Our next question is, has SoloVPE been validated for bioburden analysis, such as water, drug product, etc.?
Validated, not to my knowledge. Used for this particular application, yes. It just depends on where it is within the process. So the current companies that we work with related to this application are too far upstream and don't have GMP requirements. That doesn't mean that it ultimately can't be validated. That has nothing to do with the actual method itself. Just based on our customer feedback, we are not ready yet to share that information in the validated environment.
Okay. Our next question is there any plans to improve the audit trail of the software?
Your timing couldn't be better. We are releasing our next generation platform of software at the start of Q2 this year. And yes, it's a completely redesigned software package that's independent of Agilent Carriwin UV. And all of the feedback that we've collected over the last several years based on our current customer base have been integrated into that software package.
Terrific. Thanks, Joe. Moving on to our next question. Can you give an example how the concentration of Polysorbate 80 is analyzed in a protein formulation?
Sure. I highly advise you to read both articles that are available for download under our literature section of the website that go into more detail. A short answer is that we found, obviously, protein absorbs at 280 nanometers. But polysorbate, whether it's 80 or 20, actually has a natural chromophore at 235 nanometers. The reason why it's never been seen is it's so concentrated that you would never see it in a standard 1 centimeter cuvette. You can only see it using the pathlength resolution on the SoloVPE. So therefore, we have, again, 55 nanometers of separation between the two wavelengths of interest, and therefore can analyze how much protein and polysorbate within the same sample.
Excellent. Let's move on to our next question. Can you check oligonucleotide, i.e. primer purity? I am concerned about truncated synthesis products or oligos leaching from recycled HPLC columns.
Okay, part of the first question, the short answer is absolutely yes. Those have been more of our newer publications that have come out even within the last calendar year, 6 months. And those are primarily based around large companies that are using the technology for exactly this application. So short answer is yes. Mm-hmm. The second part, I think, would be more specific to your own process, and I need to probably chat with you a little bit further to give you a responsible answer.
Okay. Moving on to our next question. Is light scattering off of particles ever an issue, and how do you address it?
Sure. That is such a large question. I will say that the current software that we have allows you to mimic the same light scattering algorithm as most current UV applications. So whether you were using a single wavelength approach or the USP guidelines of multiple wavelengths and plotting that line, we pause a little bit because light scattering potentially, within a 1 centimeter cuvette, okay, I understand it, but we're talking about microns differences here with our technology. So typically we like to look at the actual slope contribution at the scattering wavelength of interest. So for example, if we were looking at 320 or 350 nanometers and looking for the presence of change of absorbance, that's the real way to do it using slope spectroscopy, because if you have no change of absorbance, therefore it can't interact with the measurement at, say, 280, but we never take away that functionality. So if it's part of your current method and you want to keep apples to apples, sure, we allow for that in the software. However, ideally you work with our team and for once and for all, understand if it's really something you need to be concerned about with your sample.
Mm-hmm. Okay. Our next question is, can we measure fluorescence and bioluminescence?
Yeah, unfortunately not. We're a UV technique. I would love it too, but no. So look at this as purely UV applications, a better UV, but nonetheless UV.
Mm-hmm. And then a follow-up question from the same person, is it portable for field application?
Yes, typically I'll say the most common area where our system is mobile are in manufacturing suites where they're typically installed and qualified on a cart, and then the cart can be wheeled to the process for, subsequent analysis. We don't recommend, however, if you're saying field, outside the facility, probably not. But if you're talking about internal use, sure.
Okay. Our next question is, what is the price of the instrument?
I would say contact your local sales.
Sure. Our next question is, what is the biggest value that your customers see after implementing SoloVPE?
Sure. I would say it's sort of a 1A, 1B situation. One is the obvious, it's time savings, right? So all of that time spent prepping and diluting samples, and the more downstream you get sending samples out for analysis, that's gone. So you'll see in several publications that we've written, you're talking about hours-long processes being done in minutes. So time saving is certainly number one. And number two is less investigational issues. We have removed the most error-prone issue associated with UV by removing dilutions. And now we guarantee linearity within every sample measurement. So real-time decision-making is provided to be one of the biggest benefits of using our technology.
Excellent. Thanks, Joe. Moving on to our next question. You mentioned that oligos can be measured undiluted up to 200 milligram per milliliter. What is the maximum plasmid concentration I can measure undiluted?
Yes, we've stated 200 only because, call it fortunately, unfortunately, no one has been able to give us higher concentration material to test. My feeling is we can probably approach somewhere between 250 to 300, but I don't want to formally make that statement until we've actually had a chance to test that material. Unfortunately, you can't buy that off the shelf. They just don't make samples that high in concentration to test.
Okay. Our next question is, what are the advantages to measure drug-antibody ratio on a SoloVPE compared to traditional UV?
Sure. So twofold. We're talking about multi-wavelength analysis. Okay, so we're taking potentially two actives in the same sample that vary in concentration. So we're talking about two different preps and dilutions to be read on a standard spectrophotometer. That's gone. So in 2 minutes, you're going to get slope-based data at each wavelength of interest, undiluted and proven with linearity within those given samples. The second part is that the post-calculation math to calculate the DAR is now inherently part of the method within the software itself. So there's no need to bring any post-calculations to external programs like Excel or an unvalidated platform in order to finish the calculations. It can be part of the SoloVPE method, which can be locked down and non-editable.
Okay, thanks, Joe. Our next question is it relevant to use a buffer blank in this system?
In most cases, the answer is no. The reason why is most buffers don't physically have change of absorbance. So if you were to run your buffer as you would a sample, you'd see a flat line with a very poor R-squared value. So in most cases, there's no buffer correction. The only time we would potentially recommend baseline correction or a buffer blank is if you were testing something extremely dilute, where the slope of the sample and the slope of the buffer might be similar in value. It would be best to tear one of those out. But particularly any high concentrated sample, forget the application, it's a blanket statement. Any high concentrated sample, there's no influence of buffer there, so you can remove that completely from the experiment.
Mm-hmm. Our next question is, how much calibration is needed?
It will be no different than your current method. So typically, we recommend there's a simple transmission check that you would run at the start of the week that just measures light output. That minute is a 1-minute test, and then you would run a system suit standard, comparing the slope value compared to the value on the certification document, whether you're reporting absorbance or concentration, but it's no different than your other UV technologies, just replacing slope from absorbance.
Excellent. Thanks, Joe. Our next question is, I have used the SoloVPE in different companies, one using Chem 013 standard as system suitability, the other only an internal standard. Would you recommend using the Chem 013 standard?
We use the we call it Chem 13 standard as part of our IQ, OQ, and PM maintenance. We also have a NIST traceable slope standard called Confirm, which is a caffeine and water solution, which is manufactured with a SoloVPE. Therefore, the traceable value, which is NIST, certifies a slope value. The honest answer is you can use any UV NIST traceable standard on our technology. So we've had groups that use BSA because it's cheap. The tolerance isn't great on it. However, you're literally just loading the extinction coefficient, wavelength of interest, and calculating concentration based off a slope value. So you're not limited to the Chem 13. You can use any UV NIST traceable standard you'd like.
Okay, terrific. Thank you. Just another reminder for everyone, if you have a question for Joe, please type it in the Ask a Question box on your screen, and also a reminder that there are additional resources available for download in the upper right side of your screen. And let's take a look at our next question now. Is the SoloVPE system and software fully GMP compliant? And how are the system and software qualified, and what type of support is provided for qualification?
Sure. Answer is yes. All the tools exist within the software to achieve compliance. During the IQOQ service, the software is qualified on it to ensure proper software loading within the qualification protocol itself. And then typically our applications group would then work with each company to design a plan for validation of that software as a platform within the company. And options exist anywhere from working with the IT group and answering a couple of basic questions, providing a at-cost protocol for your IT group to run. And we also offer services where we would come in and execute a full validation protocol within a few days at a cost, and that essentially covers the entire validation of the software itself. Throughout the entire process, you are supported by our team. So there's no scenario that involves us leaving you with a system without assistance.
Excellent. Our next question is, well, how long does it take for the system to be installed and validated for PD work?
Okay, so for PD, if you're talking about the installation and training, that can be done in a day. Validation honestly depends on the scope of the project, okay? Whether you're validating a range of concentrations of a product or a specific drug substance or drug product related to the Solo. So we've seen typical validations, on the order of several weeks, and we've also seen validations on, global transfer of a method, take the better part of a year. That is unrelated to our organization. So I can say that we will not be the bottleneck related to the work being done. Usually it has to do with resources demands internally by your organizations to determine how much time you can actually spend on the project and what time frame.
Okay, our next question is, what is the typical cost per sample with regard to consumables cost?
Sure. On average, it's somewhere between $3 to $4. We don't have many consumables. It's aside from the standard, it's just 2. So you're talking about the disposable sample vessel and the fibrette. So if you, which we recommend disposing of your consumables so you don't risk the introduction of any carryover into subsequent samples, you're talking somewhere in the range of between $3 to $4 a sample. However, I can assure you that that is substantially less than any time or cost spent diluting, preparing assays, calibration of scales that you were using with your traditional UV method.
Okay. Well, I believe that's all the questions we have today. We'd like to thank Joe Ferriolo for sharing his knowledge with us, and also offer a special thank you to CTech, a Repligen company, for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. Thanks for attending the webinar today, and please enjoy the rest of your day.